I'm Dr. Reena from Botany Department. I'm working as a lecturer in Government College for Women, Sector 14, Panchkula. Today, I'm going to explain you regarding the Mucor, which its common name is bread mold, and its systematic position is division Eumycota, class Zygomycetes, order Mucorales, family Mucoraceae, and genus Mucor. And this include 150 species, and its common name is bread mold. It has saprophytic mode of nutrition. That means it takes its food from the dead organic matter, and suitable conditions for its growth are favorable temperature and the moisture. So these two conditions are required for this fungus to grow, and all the fungi require these conditions. And most common habitat are the foodstuffs, which are rich in sugar, like jams, jellies, decaying vegetables, etc. And in India, there are 17 species have been reported. So a few examples are Mucor indicus, Mucor mucido, Mucor hymelis, and this Mucor mucido is also known as black mold, and it grows on the moist horse dung. And there's one of the species that is Mucor purcellus. It is pathogenic and cause bronchomycosis. No, some of the species are parasites also, and some of the species have saprophytic mode of nutrition. Now we'll discuss about the vegetative structure of this fungus. Now this mycelium grows on the substratum, and it consists of number of hyphae. They grow prostrate and erect hyphae also there. The prostrate hyphae, they develop certain branches which penetrate into the substratum and help in absorption of the nourishment from the substratum and help in fixation. And rhizoids are not present. And on the vertical hyphae, sporangia are developed which take part in asexual reproduction. Hyphae are aseptate, that means no septa formation, multinucleate. So this condition is called cenocytic. So these hyphae are called cenocytic because they contain number of nuclei, and they have small vacuoles also, and the food is stored in the form of glycogen and oil droplets. You can see the structure also. So you can see the prostrate hyphae which are growing horizontally, and they send their branches into the substratum. But rhizoids are not present. So you can see the vertical hyphae also, which bear the club-shaped structures known as sporangia. And in this picture, there's another diagram which is showing the dehiscence of the sporangium. So the spores are exposed and they are carried by wind currents, and we'll discuss about the asexual reproduction after this. Now you see here, we'll discuss about the reproduction. So two types of reproduction take place: asexual reproduction and sexual reproduction. Asexual reproduction takes place by fragmentation, production of oidea, chlamydospores, sprangiospores. But in this case, zoospores are not formed. And asexual reproduction takes place during the favorable conditions by the spores, and these sprangiospores are produced in the sporangia, and the sporangia are developed on the erect hyphae known as sprangiophores. You can see the diagram. So how this development of sporangia take place, I'll explain you from this diagram. Now you can see here that erect hyphae are there on which the sporangia are developed. First of all, the protoplasmic contents, they flow to the terminal portion of the sprangiophores, and the terminal portion become club shaped, and this developing sporangium shows two regions or zones. The outer zone is granular, contain more number of nuclei, and then dense cytoplasm is there, and food reserved is also present. Whereas the inner portion is having vacuolated cytoplasm, and in this way two zones are developed. Now later on what happens that vacuoles are developed which fuse to form a continuous cleft, and then dome shaped septum is formed towards the inner side of the cleft. You can see here in the diagram also that the dome shaped septum is formed, due to which dome shaped columella is formed, which is in continuation with the sprangiophore. So in this way the two zones are separated by the formation of septum. The peripheral dense portion show cleavage, and in this way the protoplasmic fragments are produced, which are small pieces of, or you can say fragments which contain number of nuclei. Later on these fragments they round off, they develop their own wall, and in this way sprangiospores are developed, and these are non-motile and multinucleate, and their wall is also thick. And I told you earlier also that when this fungus grow on the its substratum, it forms a white cottony mass, and later on it becomes dark in color due to the formation of reproductive structures. So in this way the spores are developed. Now next I'm going to explain you regarding the dehiscence. So you can see here in the diagram only regarding the dehiscence. So you can see here in the diagram that the the protoplasmic contents continues to flow towards the columella from the sprangiophore, and this columella increase in size and exert pressure on the spore sac. In this way the sporangium wall will rupture and these spores are exposed, and initially they are attached to some of these spores are attached to the columella, and later on they become dry and they're carried by wind currents. And in the basal portion you can see that some remains of the sporangial wall are attached like a collar. Now these spores are carried by the air, and they can also survive in the unfavorable period, and we can say that they are perinating structures. And whenever they get suitable substratum, and they get favorable temperature, moisture, which are the conditions required for the spores to germinate, then they absorb moisture and they start germinating and produce small hyphae and leads to formation of a new mycelium. So this is all about the asexual reproduction. Now next I'm going to explain you regarding the sexual reproduction. So sexual reproduction is isogamous type. Isogamous type of sexual reproduction means two fusing gametes. These are morphologically identical. So we cannot judge morphologically whether they belong to different strains. And these gametes are non-motile, and they are also known as planogametes and gametangia. And in this case, during the sexual reproduction, the gametangia fuse, and in this case, we say it as gametangial copulation. Now, in this case, the entire content of the gametangia, they fuse with each other. Now, what happens if the gametangia belong to the same mycelium? Then that means the fusing gametangia belong to the same mycelium. We say that these are homothallic species. And if the gametangia which are going to fuse, they belong to different mycelia, we call it as heterothallic species. That means there is some distinction between two different type of mycelia and which are involved in gametangial copulation. And we can say that one of the mycelium belong to the positive strain and other belong to the negative strain. Now this process of sexual reproduction, this is initiated by the formation of sex hormone. And what happens that the two mycelia which belong to the different strains, they come together and they develop special hyphae known as zygospores. Now you can see all this in the diagram also. So in the diagram you can see that one of the mycelium belong to the positive strain and other belong to the negative strain. Now these two mycelia, they come together, they develop special hyphae known as zygospores. Now zygospores come together and they develop copulating branches, and these branches are known as progametangia. Now after the formation of progametangia, you can see here in the diagram the protoplasmic contents of the progametangium, they start shifting towards the terminal position. So terminal जो portion होता है fusing gametangia का, that become club shaped, and this portion which is having dense cytoplasm and it is more granular and more number of nuclei are there and more food is reserved, this becomes separated from rest of the portion of the progametangium, and rest of the portion that contain more number of vacuoles and less number of nuclei. So this terminal portion that form the gametangium, which gets separated from rest of the portion, which is called suspensor. Now the gametangia will fuse, the contact wall will dissolve, and the two gametangia will fuse and leads to formation of xenozygote. Now why it is called xenozygote? Because it contains number of nuclei. After this, the nuclei, they don't fuse but they come together and they are arranged in pairs, and the unpaired nuclei will degenerate. Now in this way, the paired nuclei will fuse, and then in this way the diploid nuclei are formed, and later on this xenozygote enlarge, develop its two-layered wall and become zygospore. Now you see the structure of zygospore. So I told you regarding the formation of zygospore, how it happens in sexual reproduction. Now I'll explain you regarding the structure of zygospore. So you see that zygospore has two-layered wall, outer exine, it is thick and known as exospore, whereas the inner is intine, known as endospore. and this enclosed dense cytoplasm, diploid nuclei, and food is reserved in the form of glycogen and oil. Now, this mature zygospore becomes separated. How it is liberated? This is by weathering of suspensor. After liberation, this zygospore undergoes a period of rest for 5 to 9 months, and after that it will germinate whenever it will get favorable conditions. So after a period of rest, the diploid nuclei start dividing by meiosis, and sometimes the meiosis takes place before the resting period of the zygospore, and sometimes the meiosis takes place after the period of rest of the zygospore. Now, the nuclei which are produced after meiosis, four nuclei if they are produced, the two belong to the positive strain and two belong to the negative strain. Now, I told you that in this case also the nuclei except one, all the nuclei except one degenerate. So that means only one nucleus is there, and that belong to either positive strain or negative strain. Now this nucleus will undergo mitosis and produce number of nuclei. So you can imagine that the mycelium which will be produced from this will be either of positive strain or negative strain. Now I'll explain you regarding the germination of this zygospore during the favorable conditions. Zygospore absorb water, swells, and its exospore ruptures, whereas the endospore or the intine comes out or protrude out in the form of a germ tube, which is called promycelium. Now, what happens that the cellular contents of the zygospore, they transfer to the tip of the promycelium, and in this way the terminal swollen shaped structure is formed, which is called zygosporangium. Now what happens that the protoplasmic cleavage takes place, and later on uninucleate spores are produced, and later on its nucleus undergo mitosis, and in this way the multinucleate spores are formed. Now these spores, whenever they'll find suitable conditions, they can produce a new mycelium. In some cases, normal gametangial copulation do not take place. So what happens under these conditions? Either one or both the gametangia, they produce structures similar to the zygospore. So this type of zygospore, which is produced parthenogenetically, is known as azygospore. So this is all about the life cycle of Mucor. Next, so I have already explained you everything regarding the sexual reproduction. Next, I'll explain you regarding the heterothalism in Mucor. So as I've told you earlier also, what is heterothalism? So it's a condition where the thalli of single species are morphologically identical, but physiologically they are unisexual or they are different. This phenomena of heterothalism in Mucor was discovered and studied by A.E. Blakeslee in 1904. So as I told you earlier, that if the fusing gametangia belong to the same mycelium, then these type of species are called homothalic species. And if the fusing gametangia, they belong to the different thalli, this these species are known as heterothalic species. So that means the fusing gametangia belong to different mycelia, and these mycelia might be morphologically same. Of course, they are morphological same, but we say that they belong to different strains because physiologically they are different. Physiologically, one of the strain is male and other is female. So morphologically we cannot distinguish them, but physiologically they are different. And they observe that the mycelium which grow vigorously is female, or we can say the positive strain, and the mycelium which grows slowly, that is male, and that belongs to negative strain. So this is heterothalism in case of Mucor. Blakeslee, he proved this heterothalism in Mucor musudo by an experiment. What he did, he inoculated a sterile agar medium in a petri dish with many spores belonging to positive and the negative strain, and he found that spores germinate to produce mycelia. Later on, he observed that zygospores were produced when the fusing gametangia belonged to positive and negative mycelium, and zygospores were not produced when fusing gametangia belonged to either both the positive strains or both the negative strains. So that means there is some different physiologically and morphologically, although they are identical, but physiologically they are different, and one belong to positive strain and other belong to negative strain. And later on, he also did biochemical test, and he observed that positive strain mycelia grow better on maltose and have capacity to reduce tellurine salts when they were compared to the negative mycelia. So this is how we have explained about the Mucor and heterothalism in the Mucor. Thank you.